Hello everyone, myself is Dr. Avinash Ammangi. I am from Gurudev Academy, Dharwad. Today we are going to discuss with respect to the synopsis part of what biomolecule, which is related with respect to the Karnataka Common Entrance Test. So this will be the brief description what we are going to discuss with respect to the biomolecules. Right. So majorly if you consider, right, biomolecules are the chemical compounds which are actually found out with respect to the what living organisms, right? So biomolecules are the chemical compounds that found in what particularly living organisms, and they include what? They include organic and inorganic compounds too, right? Like inorganic component forms what? Organic one. Organic component get metabolized to form what? Inorganic one. Like that, what overally the biomolecule is made up of. So analysis of what chemical composition in with respect to the tissue culture, if we carry out, like what they are going to do is the grinding of what living tissues, mainly like vegetables or piece of a liver, etc. you can take, right? That to what mainly with respect to the trichloroacetic acid, right? So trichloroacetic acid, with respect to that we'll take some vegetables or like what piece of what leaves or piece of what liver you can take, right, to get what the thick slurry. Then they are going to carry out what grinding of this particularly what tissues. Now when the grinding is performed, then we are going to get something called as what this thick slurry. Then strain this through the what the particularly cheesecloth or the particularly cotton to get two fractions such as what filtrate, acid soluble pool, and the retentant, that is nothing but acid insoluble fractions. So what we are going to get when we carry out a filtration, some of the part is going to be reside there only, and some of the is going to get what? Filtrate. So filtrate is acid soluble component and retentant is what? Acid insoluble fractions, what we are going to get it. With respect to the what? Filtrate, mainly like what? Acid soluble pool contains what? Biomacromolecules, that is molecular weight is around what? Less than 1000 Dalton, right? And retentant, mainly like what we call it as acid insoluble fractions, which contains mainly the biomacromolecules, mainly like what? Molecular weight is greater than what? 1000 Dalton, right? So filtrate contains what? The particularly biomacromolecules which is less than what? 1000 Dalton in weight, and the particularly retentant or acid insoluble fraction consists of what? Biomacromolecule which is more than or greater than the what? 1000 Dalton with respect to its molecular weight, right? So like that, what we can go ahead with respect to the extracting these components, right? Then weigh a living tissue, majorly like what? Wet weight, right? And it is dried, that is what we call it as dry weight, to evaporate the water from it. Now as even what we know, the particularly our body itself is made up of like one is wet weight and dry weight, right? When water content is actually removed from the tissues or from the what cell overally, then it is going to have what something called as what? Dry weight, right? Wet weight where water is actually included in that, right? So it is fully what? Burnt to what? Oxidize all the carbon compounds to gaseous forms, right? So CO2 and water vapors, right? And it forms something called as what? Ash. So ash like structure, what we are going to find out after like what? Dry weight, when drying the component we are going to go for what? Burning, and to what? Oxidize all the carbon compounds present inside that. Then you will find out something called as what? Ash, right? Now this ash mainly contains what? The inorganic element. Majorly this whatever the ash is, if we go for a chemical analysis, it is consist of what? Inorganic element, that is calcium, magnesium, sodium, potassium, and even inorganic compounds like what? CO4 2 minus, PO4 3 minus, NaCl, and calcium carbonate. Like that of what component you are going to get it with respect to the what? This ash, right? Then overally like comparison of what element in a non-living and the particularly living matter. So which is actually there with respect to the something called as what even NCERT 2, right? Consider we take element as what? Hydrogen. Hydrogen you can find out with respect to the earth crust is what? 0.14, and human body is around what? 0.5, right? So human body content of hydrogen is what more compared to the what earth crust, right? Then carbon source if we consider 0.03, right, in particularly what with respect to the earth crust. Then with respect to the human body it is 18.5, right? Then oxygen component 46.6 you are going to get it with respect to the earth crust, and human body which is around what 65, right? Then nitrogen content you can consider, which is actually what very little you can find out, and 3.3 you are going to get it with respect to the what human body, right? Then sulfur you are going to get it is around what 0.03, and human body is around what 0.3 percentage, that much of what the particularly sulfur content you are going to find out. Then you will find out what the sodium which is around what 2.8, and human body it is around what 0.2 percentage, right? Then you consider like what calcium is what around 3.6, and with respect to the human body it is around 1.5, right? Magnesium if I consider, which is around what 2.1, and the with respect to the human body is what 0.1, right? So silicon is the more abundantly found out with respect to the earth crust, like what 27.7, comparing with respect to the human body, and it is what negligible with respect to the what majorly in human body, right? So like that of what component you are going to find out, right? So we'll go ahead with respect to the what deciding, understanding what exactly these biomicromolecules are. I'm talking regarding macromolecules. Micromolecules combines to form what macromolecules. So that itself what we are going to discuss here, right? Now if we consider majorly like what the biomacromolecules, that is molecular weight of what biomicromolecule, right? Majorly like what micromolecules or biomolecules found in the particularly what acid soluble pool. So whichever got what dissolved with respect to the trichloroacetic acid, that itself what we call it as the particularly biomicromolecule, right? So you consider what weight, consider that is what ranges from 18 to what 800 Dalton, like what we have studied before, that is less than the 1000 Dalton, whatever the weight of these molecules are, those are considered to be what biomicromolecules. The acid soluble pools represents the particularly cytoplasmic composition. Majorly acid soluble pool actually represents what the cytoplasmic component. So overallly whatever this extraction what we have performed with respect to the trichloroacetic acid, what it consists of, these majorly biomicromolecules include what amino acids. So amino acid to combine to form what protein, which is a macromolecule, lipid to form what the particularly fat, then sugars combine to form what something called as what carbohydrate, nitrogen bases combine to form what something called as what with respect to the DNA structure. That what you are going to find out. So that what we are going to discuss one by one, that is what with respect to the amino acids, right? They are the compound formed of what an amino group. It consists of what an amino group and an acidic group, right? Structurally if you find out, this is known to be what amino group. With respect to that we will find out what one the carboxylic group or the majorly carboxylic acid group, and H and A variable group what you are going to find out. So the H with respect to that R represents what variable side chain. Onto that there can what attachment of what any kind of what group you are going to find out, right? So majorly amino and what carboxyl group or carboxylic acid are attached to the same carbon atom, right? That is what alpha carbon atom. So they are called as what alpha amino acids. So with respect to the what alpha carbon, same carbon only, there will be attachment of amino group and carboxylic group. So only we call it as what alpha carbon, and they are substituted methanes. They are what majorly substituted form of what methanes. So here some of the what amino acids you consider, right? So R is what going to be variable, right? In glycine majorly what you are going to find out, consider like what remaining structure will be same, that is what C to the attachment of what amino group, and one to the attachment of what again carboxylic acid group, and one hydrogen will be a common. So here what happened, one H is attached we call it as what, the glycine structurally, right? Same way only the R group got changed, that is what, CH3 it got attached, and that is nothing but alanine. That is known to be what, alanine. Consider it is attached with respect to the CH2OH R group, that is nothing but what, known to be the what, serine. So like this, what commonly we can remember the structure of this particularly amino acids, right? Then you consider majorly there are what, 20 types of what amino acids are used in particularly what, protein synthesis, right? Amino acids are of what, three types mainly, right? So acidic amino acid, that is what, glutamic acid is an example. Then basic amino acid, that is one more example is what, lysine. Then the neutral amino acid, that is nothing but what, valine. So structurally if we find out with respect to the structure only and its nature, based on the nature only, like what they have told is acidic amino acid, example what here is, you should remember the example, glutamic acid. Then basic amino acid, that is nothing but what, lysine, and the neutral amino acid, that is nothing but what, valine. So like that what you should remember. So some amino acids are what, aromatic even, right? They are what, aromatic, structurally aromatic form of what structure, what you are going to find out. Example is what, tyrosine, then phenylalanine, and what, tryptophan. So aromatic type of amino acids examples are what, tyrosine, phenylalanine, and tryptophan. So this you should remember, right? Then amino acids can also be classified as follows, right? Mainly what we call it as essential amino acids, and one more is known to be a non-essential amino acids. Now what is this essential amino acids? They cannot be synthesized by body, right? Essential means we need it, right? So we need it means obviously we should supply it from the what, diet, right? So the body cannot synthesize that, so should be what, supplied through our what, diet, right? Examples are what, the lysine you consider, leucine you consider, isoleucine you consider, and tryptophan. With respect to the what, the dietary element, we are going to get what, all these type of what, amino acids. Those amino acids are known as what, essential amino acids. Non-essential amino acid, we no need to what, supply anything from now, so they can be what, synthesized by whom, the body itself. Body itself or the cell itself is going to form these particularly non-essential amino acids. Example are what, glycine, alanine, serine, and arginine you are going to find out. So these are the what, examples consider, right? Then again in amino acid, majorly what, the amino group or carboxylic group have a ionizable nature. They have what, ionizable nature. So structure of this particularly amino acid changes in solution of what, a different pH, right? So as these have what, both the charges, positive as well as what, negative. Amino group gives it positive and carboxylic group gives it what, negative. So it is known to be what, a neutral. But what you can consider over here is the solution wise, if I give it what, acidic type of what, solution, the particularly it will become what, acidic in nature, right? If I give it what, the particularly basic type of what nature, then it will become what, basic type. So because of that only, like if both amine and carboxylic groups are what, ionized, if they are ionized with respect to the what, different pH, it is called as what, the Zitter ion. So what is amino acids? Amino acids are nothing but they are Zitter ionic in nature or Zitter ionic in form. So that is what you should remember here. Then orally, like what we have discussed basically with respect to the amino acid, the next is what, the particularly what, lipids. Still we are with respect to the what, the biomicromolecules. That what you should remember, it is a micromolecule, right? Now these particularly lipids, consider it is water insoluble, it won't get dissolved inside the water, contains majorly like carbon, hydrogen, and oxygen, but number of what, oxygen atom is what, less. So what you should remember with respect to the lipid, the number of oxygen atom is less with respect to the what, lipid. So you consider ghee, oil, right? Or even like what, if you want to mix up with respect to the what, water, you will find out the oil is going to float onto the what, particularly water. That is what we actually call it as lipid, right? Then again types of what, lipids if you consider, that is simple form of what, lipids, then compound lipids, then derived form of what, lipids. So one by one we will understand. So simple form of lipids, compound lipids, and derived form of what, lipids, right? Then you consider majorly like what simple form of lipids, what it is actually. These are the formed of what fatty acids and alcohol, like a glycerol. So glycerol and fatty acid, if it get combined, I'll call it as what simple lipids, right? Or alcohol. Glycerol is nothing but an alcohol. So fatty acid and alcohol includes to form what simple lipids. Fatty acids are what lipids with the what hydrocarbon chain, right? And it has what R group, right? Again ending in with respect to the what carboxylic group, that is what RCOOH, right? So fatty acid of lipid consist of what RCOH. Then example what they have mentioned is what palmitic acid. So palmitic acid has what, how many carbons here? 16 carbon compound it is. So structure what they have mentioned is CH3, right? Then CH2 raised to what 14 overally COOH, right? So or we can mention it as what C15 H31 COOH, right? So and the particularly arachidonic acid, if you consider, which has what a 20 carbon compound, right? It is a 20 carbon compound, this fatty acid, and this fatty acid palmitic acid is of what 16 carbon compound, what you are going to find out. So palmitic acid is 16 carbon compound, then arachidonic acid you can find out with respect to the what 20 carbon compound. So this what you should remember. Mention something called as what 16 carbon compound fatty acid, that is nothing but palmitic acid. Mention something called as what a 20 carbon compound fatty acid, that is nothing but arachidonic acid. That is what you should remember, right? Then a type of what fatty acids here, right? Mainly, right, what we are finding out here, saturated fatty acids and even what unsaturated form of what fatty acids. Now what is saturated fatty acids here? They have what non-double or triple bonds, right? They have no what double bond or triple bonds between what carbon and carbon atoms. So between carbon atoms you are not going to find out any double bond or triple bond. They are known to be what saturated fatty acids. So example what it is, palmitic acid, then stearic acid, right? So this what you should remember, this is even made up of what 18 carbon compound, right? So that is what you should remember, that is stearic acid 18 carbon compound. Palmitic acid is 16 carbon compound, comes with respect to the saturated fatty acids, right? Which doesn't have what a double bond as well as what triple bond. Unsaturated fatty acids majorly they have what one or more double bonds. They have one or more double bonds, you are going to find out. Example they have mentioned is what oleic acid. So oleic acid is of what 18 carbon compound. This is what you should remember. Then arachidonic acid, which is nothing but what orally 20 carbon compound, right? So these are known to be what unsaturated fatty acids. So that what you should remember, right? Then structure of what glycerol, if you consider, this is the overall structure of what glycerol, that is what it is CH2OH, right? Then CHOH, right? And with respect to that CH2OH, right? Like that of what arrangement. Mainly structure of glycerol, we call it as what trihydroxypropane. It is known to be what trihydroxypropane, right? Now with respect to this, fatty acids are what esterified with the glycerol through the what ester bond, right? Forming something called as what monoglycerides, right? So diglycerides and triglycerides. When fatty acids are esterified with respect to the what glycerol through the ester bond, forming what monoglyceride, triglyceride or diglyceride, like a food structure, right? So palmitic acid structure what they have mentioned, and attachment of what glycerol you can find out, right? That is what a fatty acid which is attached with the glycerol to form what a lipid. So one glycerol plus one fatty acid known to be what monoglyceride, right? One glycerol with respect to the two fatty acids we call it as diglyceride, right? So then again one glycerol plus three fatty acids we call it as triglyceride, right? So mainly this is known to be the what monoglyceride, this is known to be the structurally diglyceride, and this is nothing but triglyceride. Attachment of what 1, 2 and 3 type of what fatty acids. So how many fatty acid attachment with respect to the glycerol? Like that only it can be divided into monoglyceride, diglyceride and triglyceride like structure, right? Then triglyceride, if you consider, which is based on what the melting point lipids triglycerides are of what? Two types, based on what? Majorly melting point. Fats has what? A higher melting point, and oils has a what? The particularly lower melting point. So fats has what? Higher melting point, right? Then the oils has a what? The particularly the majorly lower type of what? Melting points. That what you should remember. So here is what with respect to the triglyceride attachment is what? R1, R2 and R3, right? So three type of what? The particularly fatty acids are going to get combined. So compound lipids majorly like simple what we have discussed. Now we'll discuss something called as what? Compound type of what? Lipids. So these are the what? Esters of what? Fatty acids. These are what? Esters of what? Fatty acids and alcohol with additional group. Again additional group you are going to find out. Like for example phospholipid, right? So which is a majorly part of what? The structure of what? Cell membrane. So already you have discussed with respect to the what? The particularly cell, the unit of life. Inside that the particularly the Sanger and Nicholson's the particularly fluid mosaic model of what? Plasma membrane. There you have came across with respect to this particularly phospholipids. Like so majorly here you can consider fatty acid combines with respect to the glycerol and one more group known to be the what? Phosphate. So they are found in with respect to the what? Cell membrane, right? So example even with respect to the what? Phospholipid is lecithin. You should remember what example for the particularly phospholipid is lecithin, right? So that what you are going to find out. Then there is something called as what? Derived form of lipids. With respect to the derived form of lipids, so these are the what? The product of hydrolysis, right? So they are known to be the product of what? Hydrolysis of some simple lipids and compound lipids. So when compound and simple lipids get hydrolysis, it forms something called as what? This derived type of what? Lipids. So cholesterol is being one example for this. So derived lipid example is what? Cholesterol, majorly which is actually obtained from the what? Simple and compound lipids hydrolysis, right? So that's what we call it as derived lipid, as these are the product of hydrolysis of simple lipid and compound lipid. Example is cholesterol. That what you should remember, right? Then there is something called as what? Sugars, or even what we call it as carbohydrates, right? So majorly if you find out, sugars are the what? Sweet and water soluble carbohydrate. So they are formed of what? Carbon, hydrogen and oxygen in the ratio of what? 1:2:1, right? So this is what you should remember. So majorly sugars are what? Sweet and water soluble carbohydrate, and they are formed of what? Carbon, hydrogen and oxygen in the ratio of what? 1:2:1. So that is what you are going to find out. So example they mentioned it as glucose and ribose, right? Glucose is known to be the what? Hexose type of what? Carbohydrate, right? Hexose. And this is known to be the what? The particularly pentose, right? So means based on the carbon compound, that is what? C5. Here you will find out C6, right? So structurally if you find out what it is, glucose structure is C6H12O6, right? Here pentose you are going to find out C5H10O5, right? So based on the what? Structure only we call it as what? Hexose sugar, and that is known to be the what? Pentose sugar, right? Then majorly like what? Nitrogen bases, right? Even you can consider with respect to this particularly carbohydrate can be what? Divided into what? Different categories, right? So based on the category if you consider, they are known to be monosaccharide, right? Based on the what? Chain of homopolymer of what? This particularly glucose molecule, right? Then consider only one is there, only one component I am finding out, majorly like what? Glucose you consider, then glucose or fructose, that is what we call it as monosaccharide. Then there is something called as what? Disaccharide. So disaccharide majorly example you can consider what? Sucrose, right? So which is made up of two molecules of one molecule of glucose and one molecule of fructose, right? Then there is something called as what? Polysaccharide, which is majorly like what? More than what? 10 homopolymers are going to get what? Attached with each other and forms a what? Structure, a long structure. That is what we call it as majorly polysaccharide, right? So that is what you can consider. Then nitrogen base consider, which is actually a component of something called as what? Our nucleic acids, DNA and as well as what? RNA. So these are the what? Nitrogen containing cyclic compounds found out with respect to the what? Nucleic acids. So there are they are what? Classified as follows. So how they are classified? They are classified based on what? Purine and pyrimidine. So purines are what? Adenine and guanine. So pyrimidines and cytosine, thymine and uracil. So structurally you can find out this is known to be the what? Adenine. Majorly known to be the purine. This is known to be the uracil, majorly known to be the what? Pyrimidine. So nitrogen bases can be what? Divided into two, that is what? Purines and pyrimidines. Purines are nothing but adenine and guanine. Pyrimidines are nothing but cytosine, thymine and uracil. That is what you are going to find out. Then nucleoside, consider majorly like what exactly this is nucleoside. Consider one nitrogen base combines with what? Pentose sugar, right? Either this pentose sugar will be like what? A deoxyribose sugar or the particularly ribose sugar. So these are the what? Compound formed by linking the nitrogen base with respect to the sugar molecule. With respect to the what? Sugar molecule, if there is an attachment of what? The particularly nitrogen base, we call it as what? A nucleoside. So nitrogen base plus sugar forms what? Nucleoside. Consider for example the majorly adenine combines with a sugar molecule, either it is deoxyribose or ribose, so forms what? Adenosine, right? And if it forms what? Guanine to the what? Sugar, guaneosine. Then cytosine to sugar, then forms what? Cytidine. Thymine to what? Sugar, forms what? Thymidine. Then uracil to what? Sugar, uridine. So what we know the difference between what? A deoxyribose and ribose is, there is like what? In a deoxyribose only these components can be found out: adenosine, guaneosine, cytidine and thymidine. But with respect to the what? Ribose, what you are finding out? Adenosine, guanine, cytidine and what? Uridine. So instead of what? Thymidine, you will find out what? Uridine, which is nothing but known to be the what? Orally nucleosides. That what you should remember. So the structure what they have mentioned over here, right? Then the nucleotide. Now nucleotide is what? It's a combination of what? Nitrogen base, a sugar molecule with respect to that what? Phosphate group. Then only it forms what? A nucleic acid. So it creates what? Nucleic acid. So acidic nature for this particularly nucleic acid, either RNA or DNA, it is due to the what? Attachment of what? The phosphate group, right? And that attachment of phosphate group itself will create what? Nucleotide, right? Or even you can say it as what? Nucleoside plus phosphate is what? Nucleotide. And nucleoside consists of what? Nitrogen base and sugar. So that is what we have studied before, right? So these are the what? Compounds formed by what? Linking of what? Nucleoside, that is nitrogen base plus what? Sugar with what? The particularly phosphate. So nitrogen base plus sugar plus phosphate forms what? Nucleotide. Consider it is related with respect to the what? DNA or with respect to the what? RNA even. Adenine plus sugar plus phosphate forms what? Adenylic acid. Guanine plus sugar plus phosphate forms what? Guanylic acid. Cytosine plus sugar and plus phosphate forms what? Cytidilic acid. And thymine plus sugar forms what? Phosphate plus what? Thymidilic acid. Uracil, sugar plus phosphate plus what? The majorly what it forms? Uridylic acid. Like for example, if you consider these nucleotides are related with respect to the what? DNA. Then DNA means what I am going to add over here? The sugar molecule, that is nothing but what? Deoxy. So deoxyadenilic acid, deoxyguanylic acid, deoxycytidilic acid, deoxythymidilic acid. If it is related with respect to the RNA, then I'll add it as what? Ribose. So ribose uridylic acid, right? Majorly we won't add, because what? The particularly main sugar molecule is nothing but what? Pentose sugar, which is nothing but what? Ribose. Here with respect to the what? The DNA, there is one oxygen less, so only we call it as deoxyribose, right? So that is what you can find out with respect to the what? Structure of this even nucleotide too, right? So majorly with respect to the what? Nucleotide consists, right? So nucleotides are what? Known to be the heterocyclic compounds. These are known to be the what? Heterocyclic compounds. Nucleic acids like what? DNA and RNA are made up of what? Nucleotide. So majorly the molecules known to be what? DNA and RNA is actually made up of what? Nucleotide. So like this of what? Structural arrangement you can find out, which is like adenine is attached with respect to the phosphate and even with respect to the pentose sugar to form what? The particularly nucleotide. Then there we are going to discuss with respect to the what? Biomicromolecules. Biomicromolecules structurally if we consider, majorly what you are finding out over here. So these are the molecules having what? Molecular weight greater than the 1000 Dalton. That is what we have already discussed. Then they include what? The proteins, polysaccharides, nucleic acids, right? Majorly if you consider, protein is made up of what? Amino acids, that what I have already discussed. The chain of what? Amino acid forms what? Protein. Polysaccharide means what? A chain of what? This particularly glucose molecule or carbohydrate you consider. Likewise, the particularly it is known as polysaccharides. Nucleic acids, it is nothing but what? Chain of what? Nucleotide, that what you are going to find out, right? So their molecular weight is what? 1000 Dalton and above you are going to find out. They are what? Acid insoluble fractions. They are not going to get acid soluble, or they are not going to get solubilized inside what? Acid, mainly trichloroacidic acid. Macromolecular fractions you are going to find out, which includes what? Macromolecules form something called as what? Cytoplasm and organelles. So even these plays very important role with respect to the formation of what? Cytoplasm and even what? Organelles, right? But these can be formed by means of what? Biomicromolecules. When micromolecules combines to form what? Macromolecule, that what you should remember, right? Then majorly like what? Structure of cell membrane, which is actually made up of what? The lipid is not what? Strictly a macromolecule, right? As its molecular weight does not exceed what? 800 Dalton. So we cannot consider lipid as what? The particularly strictly as a macromolecule, because its molecular weight, which is actually less than what? Which doesn't exceed 800 Dalton, but it comes under the what? Acid insoluble fractions. So you should remember this, which component comes inside what? Acid insoluble fraction, that is nothing but what? Lipid. But lipid we cannot consider it as what? Macromolecule. We should consider it as a micromolecule, because whose weight doesn't exceed more than what? 800 Dalton. So this is what the most important point you should remember, because many lipids are arranged into the what? Structure, like with respect to the what? Cell membrane. So structural formation related, there you will find out what? Many form of what? Lipids present. When a tissue is grinded, cell membranes are what? Broken. So you usually what? When tissue is grinded, cell membrane is going to get broke down and form something called as water insoluble vesicles. They cannot be what? Filtered along with respect to the what? Acid soluble fraction. So these are not going to filtered with respect to the what? Acid soluble substances. So you should remember, lipids are majorly known to be the what? Comes up with respect to the what? Acid insoluble fractions, right? But they are not the particularly macromolecule, they are the micromolecules whose weight doesn't exceed more than what? 800 Dalton. So that is the most important point you should remember. So average composition of what? Cell, if you consider, cell is made up of what? 70 to 90% of water, and protein content if you find out 10 to 15%, carbohydrate is 3%, nucleic acid you will find out 5 to 7%, ions you can consider 1%, like that of what arrangement you are going to find out. Then orally we'll go on discussing now with respect to the something called as what? Proteins now, right? Which is known to be the biomacromolecule. So now if you find out proteins are what? Heteropolymer of what? Amino acids. So you should remember this also. Heteropolymer is nothing but it's a polypeptide chain of what? Amino acid, right? So that is which is related with respect to the what? Protein. So they are polypeptide, right? Linear chains of what? Amino acids linked by what? Peptide bond. So one amino acid attaches with respect to the another amino acid with respect to what? The peptide bond, right? Then peptide bond is formed when carboxylic group of what? One amino acid react with ammonia group, right? And group of what? Next amino acid by releasing a molecule of what? Water. Dehydration occurs. So when there will be a peptide bond, what will happen? One majorly like what? Carboxylic group of one amino acid react with the one more amino acids amine group, right? So definitely there will be a like what? Loss of water, or it releases the water. That is what we call it as dehydration procedure is going to take place. So like this, the particularly what? Chain of what? Amino acid you are going to find out, which are actually attached by means of what? Whatever this particularly attached by means of what? Peptide bond, right? Then again like what? Functions of protein if you consider, which plays very important role for what? Growth and tissue repair, right? Even transport the nutrient across the what cell membrane, there also it plays very important role. Example of what protein what they have given is GLUT4, right? Enables what glucose transport into the cell. You should remember these examples too. GLUT4 enables what glucose transport inside the cell. Even it can act as what intercellular ground substances. Who is going to act as intercellular ground substances? Protein. Example is what? Collagen. Example is what? Collagen. And act as what? Antibodies to fight infectious organisms. So antibodies like of structure, those are even known to be the what? Proteins. So which will fight against what? Infectious form of organisms or pathogens, what we are going to call it as. Even it act as what? Receptors, mainly like what? Receptors of smell, taste and even hormones. That what you are going to find out. So they even act as what? Receptors. So some are known to be the what? Hormones. Example what? Insulin, right? Then enzymes, right? That is nothing but what? Trypsin you consider with respect to the what? Digestion. Then pigments, that is nothing but what? Hemoglobin. So these are known to be the what? Structural form of what? Proteins, what we are going to find out, right? Then proteins, majorly if you find out what, you should remember these two names. Most abundant protein in animal world, with respect to the animal, most abundant protein is what? The collagen, right? That is what you should remember. And most abundant protein found in particularly whole biosphere, including what? The animals too, right? Plant and animals too, whole biosphere. Which is the most abundant protein? That is nothing but what? Ribisco. And ribisco is nothing but what? Ribulose bisphosphate carboxylase and oxygenase, which is an enzyme which help in what? Fixation of what? Carbon, and that carries something called as what? C3 pathway. That is what you should remember. This is an enzyme, right? Then structural level of what? Protein, if you consider, it is made up of what? Primary structure, secondary structure, tertiary structure and quaternary structure. So based on the structure levels, these are what are divided into primary, secondary, tertiary and quaternary. We will discuss what these are, right? So majorly if you consider primary structure, what it consists of, like it describes the particularly sequence of what? Amino acids, that is positional information in with respect to the what? Protein. So it is what? Sequence of an amino acid, one after the another, positional information what you are going to find out with respect to the protein. So left hand consists of what? First amino acid, which is known as what? Left side, right? So left side whatever is there, now consider this is a left side, so that I am going to call it as N terminal, right? Or N terminal amino acids. And with respect to the right hand side, that is nothing but what? Last amino acid is known to be what? C terminal amino acids. So right hand side it is what? C terminal, and in left hand side it is made up of what? N terminal. Majorly it is formed by means of only peptide bond. So it can get what? Dissolve easily, and even help in with respect to the what? Metabolic activity, and which that metabolic activity which will help in production of what? Energy too. Then there is something called as what? Secondary structure of what? Protein. Here the one or more polypeptide chains are what? Folded in the what? Helical form. So like this of what? Helical form structure you are going to find out, right? Like this helical structure you are going to get it, right? And it has only what? Right handed helix. So right handed side, only one direction only you are going to find out. So example what they have mentioned here, keratin and even fibroin, right? Which is nothing but what? Silk fiber. Fibroin or keratin, that what you are going to find out. So here with respect to the secondary, what I am finding out, the beta plated sheet which has only one type of what? Helix, and even what? Alpha helix, that is what? The structurally you are going to find out like this, right? Which is known to be the secondary structure of what? Protein. Right, now this majorly secondary type of what? Structure of protein, majorly what it is made up of, it consists of what? Now hydrogen atom or hydrogen bond with respect to that, right? So fibrino silk protein, that is what an example you can find out. So anti-parallel beta sheet rich in with respect to the what? the alanine and what glycine, right? And small side of chain allows what close packaging. So overally alanine and glycine you can find out, right? One side you will find out what the particular alanine, one side you will find out what glycine. So that is what you are going to find out, and forms what the beta plated sheet, like this of what the beta plated sheet you are going to find out. Here you can consider strong links with respect to the disulfide bond and weak links with respect to the saline and hydrogen, what you are going to find out. So keratin, which is a typically strand of what hair, the particularly with respect to the hair you will find out this keratin type of what the protein, right? Then there is something called as what structure, tertiary structure of this particularly what proteins, right? So here if you find out what, here the helical polypeptide chain is further folded like a hollow woolen ball, right? So it is going to form a woolen ball, right? So hollow means what, little gap in between with respect to the word woolen ball, like that of what structure it can form. And it gives mainly what 3D view, whatever is the structure you are seeing known to be what 3D view. So structure, tertiary structure of is necessary for what many biological activities of what protein, here mainly like what myoglobins and enzymes. So myoglobin and enzymes, these are majorly known to be the what examples of something called as what tertiary structure of what protein, and which will help in what the many biological activities, right? So that what you should remember. Then there is something called as what quaternary structure of this particularly protein. Here you will find out what more than one polypeptide chain forms what tertiary structure, and each chain functions as what the subunit of what particularly what protein. So many structures like what beta alpha. So these came together, these two came to, four of them came together, mainly the tertiary proteins came together to form a quaternary structure. So example is what hemoglobin, what they have given, and it has what four subunits, like what 2 alpha subunits and 2 beta subunits, which are actually to be what joined to form something called as what quaternary structure. So tertiary structure, four molecules of what the particularly tertiary structure combine to form what something called as what the particularly alpha and beta sheeted plates, what you are going to find out, right? Then you will find out what protein with respect to the glands, you will find out primary, secondary, tertiary and quaternary type of what assemblies you are going to find out. So in primary you will find out it's a simple structure, in secondary you will find out beta plated sheet and alpha helix, in tertiary you are going to find out what polypeptide chain, and even quaternary you will find out assembled of what subunits, right? Then you consider overally this is what one more glance you are going to find out, right? So overall structure of this particularly protein, so you should remember this, right? Then polysaccharides, majorly you can consider these are the what polymer of sugars. Monosaccharide example is known to be what starch. Homopolymer of what glucose you are going to find out. Cellulose homopolymer of what again glucose. Then glycogen homopolymer of what glucose. Unilin homopolymer of what fructose majorly. So homopolymer of fructose is what unilin, and remaining all of them are what the particularly homopolymer of what glucose. So based on the presence of what the particularly glucose molecule only, we are going to divide into in different categories. So mainly if you consider polysaccharide, these are the what complex polysaccharide formed by means of what amino sugar, glucosamine or N-acetyl glucosamine, etc. So like that of what the particularly polysaccharide, what you are going to find out. Then chitin is what homopolymer of what N-acetyl glucosamine, and it is in exoskeleton of what arthropod and fungal cell wall, what you are going to find out the presence of what N-acetyl glucosamine. So with respect to the what this particularly exoskeleton of this particularly what some of the arthropods you are going to find out, right? Then glycosidic bond with respect to the word polysaccharide, we are going to get it. So it is the bond formed when individual monosaccharide are linked between what two carbon atoms by dehydration. Again here water is removed. After removing only, what will happen, two glucose molecule linked with respect to the what glycosidic linkage and form something called as what the particularly polysaccharide, right? So that what you are going to find out. So diagrammatic representation of what portion of what glycogen you consider. So this is what the portion of what glycogen you are going to get it, the structural way, right? So likewise, if you consider majorly starch forms what? Helical secondary structure, and it can hold what? I2 molecule, iodine molecule in the particularly helical portion, giving what? Blue color. So blue color type of what solutions what you are going to find out with respect to the starch. At it has a socket, in between of the socket it can hold up something called as what? Iodine, right? So with respect to the what? Iodine, what I am finding out what? The majorly it gives what? Blue color when it comes to the starch. Then cellulose has what? No complex helix majorly, and therefore cannot what? Hold iodine. So like that of what structure you can find out with respect to the what? Polysaccharide, right? Then remaining of this particularly chapter we will discuss with respect to the next video, with respect to the next part. So at present, thank you so much. Please do subscribe our channel and like and share.